Hello everyone. We are starting with the chapter number 10, Microbes in Human Welfare. This is the first video of the chapter, class 12, Biology. In this video, we are going to cover the topics. First of all, we will do the introduction of the topic, and then we'll study about the microbes in household products, microbes in industrial products, and microbes as biofertilizers. Microbes. Microbes are present everywhere. They are present at sites where no life exists. For example, we can see them inside the geysers, where the temperature is as high as 100 degrees Celsius. We can find them deep in soil. We can see them under the layers of snow and also in the acidic environment. Microbes are diverse. You can find variety of microbes. For example, there are protozoans, bacteria, fungus, viroids, virus and prions. Prions are nothing but the proteinaceous infectious agents. They are made up of proteins and they are highly infectious. They cause diseases. Microbes like fungus and bacteria can be grown in media to form colonies, and that can be seen with the naked eyes. Such cultures are very useful in studies on microorganisms. Let us see some diagrams of various microbes. These diagrams are given in your NCERT book and we cannot avoid them. We have to see them. First, bacteria. This is a rod-shaped bacteria showing in the slide and which is 1500 times magnified. Second, this is also bacteria which is spherical in shape and it is again 1500 times magnified. Third diagram, it is a rod-shaped bacteria showing flagella. See the flagella, which are the thread-like structures, and here these bacteria are 50,000 times magnified. So actually the bacteria are of very small size. See the viruses now. First virus, bacteriophage. Bacteriophage are the virus which attack bacteria. The diagram shows it has got the head, collar, tail, plate, pins and prongs. With the help of these prongs, these bacteriophage attach to the bacteria. Second virus is adenovirus, which cause respiratory infections. Third is rod-shaped tobacco mosaic virus, and here the magnification is about 1 lakh to 1,50,000 times. So the viruses are so small. Here you can see the petri dishes. In first petri dish there is a colony of bacteria growing in it, and in second petri dish fungal colony is growing in the petri dish. So by growing them in the laboratory we can study about their behavior, about their nature. In chapter 8 we have already studied that microbes cause many diseases. वो हम पढ़ चुके हैं चैप्टर 8 में. बहुत सारी diseases हो जाती हैं microbes से, but in this chapter we will study about the uses of microbes in human welfare, their contribution in human welfare. First of all, let us see microbes in household products. Example number 1, Lactobacillus, which is also called as lactic acid bacteria or LAB. How it is useful? It converts milk to curd. How? by producing acids, and what these acids do, they coagulate and partially digest the milk proteins and milk is converting into curd. अगर हमें अपने घर के अंदर मिल्क को कर्ड में कन्वर्ट करना है तो what we do, we just add a small quantity of curd into the milk containing Lactobacillus bacteria. So why do we convert milk into curd? Why can't we take directly milk? Because in curd the amount of vitamin B12 increases. And second use क्या है इसका? In stomach, these bacteria help to check pathogens. अगर हमें कोई stomach infection हो गया है तो we used to take curd. Why? क्योंकि जो lactobacillus बैक्टीरिया हैं वो स्टमक में पैथोजेंस को की ग्रोथ को रोकते हैं, उनको चेक करते हैं. So lactobacillus are very useful bacteria. Second, bacterial fermentation. What do you mean by this? We are fermenting the dough by using bacteria. In fermentation, there is an anaerobic respiration takes place. Without air, without oxygen, bacteria are respiring. And this dough which we get after fermentation is used for making dosa and for making idli. और ये जो डो मिलता है इसकी बहुत पफ्ड अपीयरेंस होती है, फूला हुआ लगता है. वाई? बिकॉज ऑफ द प्रोडक्शन ऑफ कार्बन डाइऑक्साइड गैस. use the yeast. Name of the yeast is baker's yeast and scientific name is Saccharomyces cerevisiae. How it is useful? कहाँ यूज़ हो रहा है ये? It is used for making bread by fermenting the dough. डो को फर्मेंट कर दीजिए ईस्ट से और ब्रेड बना लीजिए, बहुत लाइट और अच्छा ब्रेड बनेगा. नेक्स्ट, toddy. It is a drink used in southern India. And how this drink is made? This is made by fermenting sap from plants. Palms के trees का जो sap है उसको निकाल के अगर ferment किया जाए, then we get the drink called as toddy drink. These microbes are also useful for fermenting the fish. See the diagram. Fermented fish is there, fermented bamboo shoot is there, fermented soya bean is there. And moreover, microbes are also used for producing cheese. The nature and the texture of the cheese are different. Flavor is different. How this is different? In Swiss cheese, we can see large holes because of the production of large quantity of carbon dioxide. Who is producing this carbon dioxide? Propionibacterium shermani is a name of the bacteria which produce large quantity of carbon dioxide and help in making the Swiss cheese which has got the large holes. Second is Roquefort cheese. The Roquefort cheese is ripened by growing a fungus. The name of the fungus is Penicillium roqueforti on them, and due to which it gives a particular flavor to the cheese. Okay. So that there are different types of cheese we can make by using different microbes. Come to the microbes in industrial products. Production of beverages and antibiotics on an industrial scale requires growing microbes in very large vessels, and these vessels where the microbes are growing in large number are called as fermenters. First of all, let us see fermented beverages. Saccharomyces cerevisiae is also known as brewer's yeast, which is used in the production of beverages by fermenting malted cereals. malted cereals are the germinating cereals and also fruit juices. Fruit juice को malted cereals को ferment किया जाए by using the yeast, then we get the ethanol. Okay, wine and beer are produced without distillation of fermented broth, but whiskey, brandy, rum, gin, arrack are produced by distillation of fermented broth. What is distillation? It is the process in which evaporation and condensation goes side by side. So, we are doing distillation. If we do the distillation, we get of the fermented broth, we get whiskey, brandy, rum, and if we do not do any distillation, then we get the wine and beer. These are the fermented beverages. Come to the antibiotics. What are anti means against, biotic means life. Antibiotics are the chemicals produced by some microbes. कुछ microorganisms एंटीबायोटिक बनाते हैं, कुछ केमिकल बनाते हैं और इन केमिकल्स का यूज़ हम दूसरे हार्मफुल पैथोजेनिक माइक्रोब्स को मारने के लिए, किल करने के लिए, उनकी ग्रोथ को रोकने के लिए इस्तेमाल करते हैं. These antibiotics are used to treat plague, whooping cough, diphtheria, leprosy, etc. The first antibiotic was made or discovered by the Alexander Fleming and it was penicillin. He observed that staphylococci bacteria could not grow around a mold Penicillium notatum growing in unwashed culture plates. Very important. अनवॉश्ड कल्चर प्लेट थी जहाँ पर स्टेफिलोकोकस बैक्टीरिया ग्रो हो रहे थे. बाई चांस वो प्लेट्स जो थी वो अनवॉश थी और अनवॉश होने की वजह से वहाँ पर मोल्ड ग्रो हो गया, पेनिसिलियम नोटेटम ग्रो हो गया. एंड इन दैट एरिया वेयर द पेनिसिलियम नोटेटम वॉज प्रेजेंट, स्टेफिलोकोकस वर नॉट एबल टू ग्रो. तो क्या हुआ? इट मीन्स पेनिसिलियम नोटेटम कुछ केमिकल्स सिक्रीट कर रहा है, बना रहा है, जो स्टेफिलोकोकस की ग्रोथ को स्टॉप कर रहा है या रोक रहा है. एंड दैट केमिकल वॉज लेटर ऑन एक्सट्रैक्टेड एंड इट वॉज कॉल्ड पेनिसिलिन. ओके, so this was the discovery of the penicillin. Later, Ernest Chain and Howard Florey, they established its full potential as an effective antibiotic. बिल्कुल क्लियर बता दिया कि हाँ, yes, we can use this penicillin as an antibiotic, and this antibiotic penicillin was used during Second World War for treating the American soldiers. And in 1945, Fleming, Chain and Florey were awarded Nobel Prize for this discovery. Come to next, chemicals, enzymes and other bioactive molecules. Let us see the organic acids first. Acid produces microbes. कौन से हैं? There is a table in front of you showing the microbe and the organic acid produced by them. The first microbe is Aspergillus niger, which is a fungus. You can see the diagram also. First diagram, and it produces citric acid. Acetobacter aceti is a bacterium which is producing organic acid called acetic acid. There is a microbe Clostridium butylicum, a bacterium which is producing butyric acid, and there is a microbe Lactobacillus which is a bacteria producing an acid, lactic acid. So, various organic acids are made from microbes, from different microbes. You have to remember the names of microbes as well as the organic acid produced by them. Next, alcohol is produced by Saccharomyces cerevisiae, and this alcohol is ethanol which is produced by this. Next, enzymes. Certain enzymes like lipases, These are used in detergent formulation. And what they do? They help to remove the oily stains on the shirts, on the clothes. See, here is a diagram. The shirt is having a spot on it, oily spot, and detergents are used to clean this. And because they contain lipase enzyme, which will digest, which will remove the oils. Next enzymes are pectinases and proteases. They are used to clarify bottled juices. See, these are the bottled juices, but normal juice जब हम घर में बनाते हैं, you can find a pulp in it all. It is not very clear, but when it is bottled in the market, then they are treated with the enzymes pectinases and proteases to clear them, to make them clear. Next enzyme, streptokinase. It is produced by streptococcus and used as a clot buster. What do you mean by this? डायग्राम देखिए, यहाँ पर हार्ट का डायग्राम है और हार्ट में एक पोर्शन ब्लैक हो गया है. इट मीन्स देयर इज अ हार्ट अटैक एट दिस पोर्शन, और यू कैन से मायोकार्डियल इन्फाक्शन. वाई देयर इज अ हार्ट अटैक? बिकॉज देयर इज अ ब्लॉकेज इन द आर्टरी. देयर इज अ क्लॉट इन द आर्टरी. द पैसेज इज ब्लॉक्ड. To remove this blockage, to remove this clot, this streptokinase are used. So they are the clot busters. Next, cyclosporin A. These are some bioactive molecules. Cyclosporin A. It is produced by Trichoderma polysporum. It is a fungus. और ये कहाँ use होता है? It is used as an immunosuppressive agents in organ transplant patients. जहाँ भी पेशेंट्स को ऑर्गन ट्रांसप्लांट किया जाता है तो उन पेशेंट्स का इम्यूनिटी सिस्टम को लो रखने के लिए, सप्रेस रखने के लिए, ताकि वो अपने आप जो ऑर्गन्स को डैमेज ना करना शुरू कर दें जो हमने ट्रांसप्लांट किया है, उनको मेडिसिन दिया जाता है साइक्लोस्पोरिन, विच हेल्प्स इन इम्यूनोसप्रेसिव, विच आर एक्टिंग एज एन इम्यूनोसप्रेसिव एजेंट्स. Next, statins. They are produced by Monascus purpurus. ये yeast है एक, और what it does? It is used as blood cholesterol lowering agents. Blood में cholesterol level को कम रखता है. Why? How? Because it inhibits the enzymes responsible for the synthesis of cholesterol. जो enzyme cholesterol की synthesis में help करते हैं उनको इनहिबिट कर देता है ये statins. So there is a question in front of you. There is a table. You have to identify A, B, C, D, E and F. And there are 3 columns. In first column organisms are there. In second column bioactive molecule is there. And in third column what is the use of it you have to write. So Monascus purpurus is the organism. बायोएक्टिव मॉलिक्यूल क्या बनता है इससे? स्टैटिन. यूज क्या है? इट इज यूज्ड एज अ ब्लड कोलेस्ट्रॉल लोअरिंग एजेंट. सी एंड डी यूज में लिखा हुआ है एंटीबायोटिक है, तो ऑर्गेनिज्म कौन सा होगा? पेनिसिलियम नोटेटम. बायोएक्टिव मॉलिक्यूल कौन सा होगा? पेनिसिलिन. थर्ड पार्ट में देखिए इसका, बायोएक्टिव मॉलिक्यूल में कॉलम फिल है साइक्लोस्पोरिन ए, तो ई में क्या आएगा? ट्राइकोडर्मा पॉलीस्पोरम इज द ऑर्गेनिज्म. और इसका यूज क्या होगा? इट इज यूज्ड एज एन इम्यूनोसप्रेसिव एजेंट इन ऑर्गन ट्रांसप्लांटेशन. 3 मार्क्स का क्वेश्चन है एंड वेरी स्कोरिंग. नाउ कम टू माइक्रोब्स एज बायोफर्टिलाइजर्स. ओवरयूज ऑफ केमिकल फर्टिलाइजर्स आर कॉजिंग पॉल्यूशन. अगर हम बहुत ज़्यादा केमिकल फर्टिलाइज़र का इस्तेमाल करते हैं तो डेफिनेटली पॉल्यूशन हो रहा है. और इस प्रेशर से, इतना प्रेशर अब हमारे ऊपर पड़ चुका है कि वी हैव टू स्विच ओवर टू ऑर्गेनिक फार्मिंग. एंड इन ऑर्गेनिक फार्मिंग वी हैव टू यूज द बायो फर्टिलाइजर इंस्टेड ऑफ केमिकल फर्टिलाइजर्स. वॉट आर बायो फर्टिलाइजर्स? Biofertilizers are organisms that enrich the nutrient quality of the soil, जो न्यूट्रिएंट्स का अमाउंट सॉइल में बढ़ा देते हैं। एग्जाम्पल क्या है इनके? बैक्टीरिया, फंगस एंड साइनोबैक्टीरिया। लेट अस सी देम वन बाय वन। बैक्टीरिया का एग्जाम्पल है राइजोबियम बैक्टीरिया, डायग्राम में भी है। राइजोबियम बैक्टीरिया कहाँ प्रेजेंट होते हैं? दे आर सिम्बायोटिक बैक्टीरिया प्रेजेंट इन द रूट नोड्यूल्स ऑफ लेग्यूमिनस प्लांट। सी द फर्स्ट डायग्राम। रूट्स देखिए, रूट्स में ये लेग्यूमिनस प्लांट्स के रूट्स हैं जिसमें नोड्यूल्स हैं और इस नोड्यूल्स में राइजोबियम बैक्टीरिया होते हैं। व्हाट दे डू? दे फिक्स द एटमॉस्फेरिक नाइट्रोजन। व्हाट डू यू मीन बाय द टर्म फिक्स? They convert atmospheric nitrogen into nitrates and bring it to the soil. अब नाइट्रोजन नाइट्रेट्स में कन्वर्ट हो गया, नाइट्रेट सॉइल में आ गए एंड दीज आर यूज्ड बाय द प्लांट्स फॉर मेकिंग द फूड, फॉर मेकिंग अदर थिंग्स. This is the symbiotic bacteria, but there are some free living bacteria also in the soil which help in increasing the nitrogen content of the soil, जो नाइट्रोजन कंटेंट को बढ़ाता रहता है सॉइल के. एग्जाम्पल है Azospirillum and Azobacter. These are free living bacteria. Come to fungus, Mycorrhiza. This is the symbiotic association of fungus, example glomus, with plants. दोनों का symbiotic relationship है, एक दूसरे के साथ मिलकर रहते हैं. फंगस को क्या मिल रहा है प्लांट से? द फंगस इज गेटिंग फूड फ्रॉम द प्लांट. तो प्लांट को क्या मिल रहा है? बिकॉज दिस इज अ सिम्बायोटिक रिलेशनशिप, तो फंगस मस्ट बी गिविंग समथिंग टू द प्लांट. फर्स्ट, इट हेल्प्स इन द एब्जॉर्प्शन ऑफ फॉस्फोरस फ्रॉम द सॉइल एंड पासेस इट टू द प्लांट. फॉस्फोरस को एब्जॉर्ब करता है सॉइल से और प्लांट को दे देता है फंगस. सेकेंड, गिव रेजिस्टेंस टू रूट बॉर्न पैथोजेंस, यानी ऐसे प्लांट जिसमें फंगस रूट्स के साथ है, रूट्स में है, वो उन प्लांट्स के अंदर पैथोजेंस नहीं लगते रूट्स में और उनकी टॉलरेंस सैलिनिटी के लिए भी बढ़ जाती है और ड्रॉट के लिए भी बढ़ जाती है एंड मोरोवर प्लांट की ग्रोथ और डेवलपमेंट भी इंक्रीज हो जाती है. नाउ कम टू साइनोबैक्टीरिया, ब्लू ग्रीन एल्गा. दे आर द ऑटोट्रॉफ्स. They can make their food. What they do? They fix atmospheric nitrogen. Again, fixing का meaning यही है, convert the nitrogen into nitrates into the soil. Nitrogen is present in the air and nitrates are present in the soil. Example of some cyanobacteria are Anabaena, Nostoc and Oscillataria. In paddy fields, साइनोबैक्टीरिया सर्व एज बायोफर्टिलाइजर्स. बहुत इंपॉर्टेंट है पैडी फील्ड्स के लिए ये. एंड व्हाट दे ऑल्सो डू, दे ऑल्सो एड ऑर्गेनिक मैटर टू द सॉइल एंड इंक्रीज इट्स फर्टिलिटी. सो हेयर वी आर यूजिंग बायोफर्टिलाइजर्स टू इंक्रीज द न्यूट्रिएंट वैल्यू ऑफ द सॉइल. सो फॉर टुडे it is complete for today. Now do the test which is given in the box in the form of an MCQs. Subscribe the channel for getting the information about the new video. Thank you very much for staying with me. Thank you so much.